Irreversible modifications on proteins accumulate during aging, leading to a decline in cellular function. The most widely studied oxidative stress-induced modification is the formation of carbonyl derivatives. Carbonyl formation occurs through direct oxidation of amino acid side chains and oxidation-induced peptide cleavage. Several systems that generate oxygen-free radicals catalyze the oxidative modification of proteins. Oxidation contributes to the pool of damaged proteins, which increases in size during aging and in various pathological states. This accumulation of modified proteins can ultimately contribute to senescent decline. In addition to generating oxidants, metabolism can produce other byproducts, including glyoxal and methylglyoxal. These trigger the formation of advanced glycation end products, which disrupt protein function and contribute to the aging phenotype. During physiological processes, proteins can also suffer additional modifications, such as racemization, isomerization, and deamination. One of the primary causes of age-related damage accumulation is the limited capacity of cellular maintenance, repair, and turnover pathways. The rate at which a protein pool is refreshed is determined by turnover, the balance between protein synthesis and protein degradation. Protein synthesis provides fresh proteins, while protein degradation removes existing and potentially damaged protein molecules. Alterations in protein synthesis occur during embryonic development, cell growth, cell differentiation, and aging. Both global and specific protein synthesis are markedly affected by aging in organisms as diverse as yeast and primates. Signal transduction cascades, such as the insulin growth factor 1, IGF-1 pathway, the kinase target of rapamycin, TOR pathway, and the p38 mitogen-activated protein kinase, MAPK, pathway, play a key role in the global control of protein synthesis by targeting several components of the translation machinery. For example, a variety of agents that promote cell growth and proliferation, including hormones, growth factors, and nutrients, have stimulatory effects on protein synthesis. Understanding these pathways is crucial for aging researchers seeking to manipulate protein synthesis for longevity. Protein synthesis proceeds through three distinct phases: initiation, elongation, and termination, which are tightly coordinated. mRNA translation initiation involves several eukaryotic initiation factors, EIFs, which orchestrate the formation of the 43S pre-initiation complex, 43S-PIC, on the mRNA being translated. This complex incorporates the initiator methionine tRNA, MET-tRNA, bound on EIF2 and joins with the 60S ribosomal subunit at the ATG start codon to form the 80S initiation complex, 80SIC, releasing the translation initiation factors. The 80S ribosome then commences elongation of the polypeptide chain. Two eukaryotic translation elongation factors, EEFs, participate in the process. EEF1 supplies the ribosome with the appropriate amino acid-loaded tRNAs, and EEF2 mediates translocation of the ribosome along the mRNA. EEF2 is regulated by the calcium calmodulin-dependent EEF2 kinase, EEF2K. Specific amino-acyl tRNA syntheses load tRNAs with their cognate amino acids. Upon encountering a stop codon, mRNA translation is terminated. The eukaryotic release factor, ERF, mediates dissociation of the ribosome from the mRNA and the release of the two ribosomal subunits, 40S and 60S. Control of global mRNA translation is mostly exerted at two crucial steps during initiation. First, the activity of EIF2, which loads the 43S PIC with methionine tRNA, is regulated by phosphorylation of its alpha subunit. Phosphorylation interferes with the ability of the guanine nucleotide exchange factor, GEF, EIF2B, to recycle GDP for GTP on EIF2. Second, the recruitment of the 43S PIC on the 7-monomethyl guanosine cap at the 5' end of all nuclear mRNAs is regulated by the cap-binding protein EIF4E. The activity of EIF4E is modulated by direct phosphorylation and/or by association with the EIF4E binding protein, 4EBP. 4EBP sequesters EIF4E and limits its availability. Phosphorylation of 4EBP releases EIF4E, which associates with the scaffold protein EIF4G, EIF4A, EIF4B, and the poly-A binding protein PABP to promote PIC assembly and protein synthesis. Understanding these intricate steps is essential for aging researchers aiming to manipulate protein synthesis. Insulin growth factor 1, IGF-1, signaling through the insulin receptor, IRS, results in the activation of phosphatidylininositol 3 kinase, PI3K, which converts phosphatidylininositol 4,5-bisphosphate, PIP2, to phosphatidylininositol 1,4,5-trisphosphate, PIP3. Phosphatase-intensin homolog, PTN, antagonizes the PTNIS3 kinase by degrading PIP3 to PIP2. PIP3, either directly or through the 3-phosphoinositide-dependent protein kinase, PDK, activates the serine threonine protein kinase ACT, AKT8 virus proto-oncogene ACT, also known as protein kinase B, ACT-PKB. ACT-PKB functions to activate the TOR kinase, either directly or through inhibition of the tuberous sclerosis complex 2, TSC2, tumor suppressor, a negative regulator of the small GTPase DREB, RAS homolog enriched in brain, which activates TOR. Upon activation, TOR phosphorylates the eukaryotic initiation factor 4E binding protein, 4EBP, which inhibits protein synthesis by binding and blocking the eukaryotic translation initiation factor 4E, EIF4E, a key regulator of mRNA translation initiation. By contrast, phosphorylation of 4EBP releases EIF4E and stimulates protein synthesis. EIF4E and eukaryotic translation initiation factor 4G, EIF4G, a scaffold protein which recruits other essential mRNA translation initiation factors, are targets of the mitogen-activated protein kinase interacting kinase, MNK1. MNK1 activity is under the control of P38, mitogen-activated protein kinase, MAPK, and the extracellular signal-regulated kinases 1 and 2, ERK1 and ERK2, also called P44 and P42 respectively. ERK1 and ERK2 respond to IGF-1 signaling and, in addition to MNK1, also stimulate the S6 kinase, S6K, which phosphorylates the small ribosomal subunit, S6, the alpha subunit of the eukaryotic translation initiation factor, 2, EIF2, which loads the 43S ribosomal pre-initiation complex with the initiator methionine tRNA, is phosphorylated by the general control non-derepressible 2, GCN2, kinase and pancreatic endoplasmic reticulum EIF2 alpha kinase, PERK, under conditions of low amino acid availability or endoplasmic reticulum stress, respectively. The amino acid transporter AAT, which imports amino acids into the cytoplasm, also signals to the TOR kinase through the TSC1, TSC2 complex and REB. EIF2 recycling during successive rounds of mRNA translation initiation is mediated by the eukaryotic translation initiation factor 2B, EIF2B, a guanine nucleotide exchange factor. EIF2B activity is regulated by the serine threonine protein kinase GSK3, glycogen synthase kinase 3, which is under the control of the AKT-PKB kinase. Although the fidelity of mRNA translation does not appear to deteriorate during aging, numerous studies have established that general protein synthesis rates decline with age in a variety of organisms. Mitochondrial protein synthesis activity also diminishes markedly during aging. While the rate of bulk protein synthesis and the activity of key mRNA translation factors diminish with age, it is not clear whether these alterations are a consequence of the aging process or whether they contribute to senescent decline. Furthermore, Different tissues and cell types are characterized by dissimilar protein synthesis activity, and tissue-specific alterations have been reported. General and specific degradation pathways, such as the proteasome system, autophagy, and lysosomal degradation, remove damaged non-functional proteins. Biochemical studies and microarray expression profiling correlate lowered protein degradation with aging and senescent decline. The activity of the cytosolic proteasomal system declines dramatically during postmitotic aging and proliferative senescence in human fibroblasts, resulting in increased half-life of oxidized proteins. Aging also perturbs 26S proteasome assembly and function in Drosophila. Interestingly, oxidized proteins and lipids, for example lipofuscin and ceroid, which directly inhibit the proteasome, accumulate during aging and thereby further exacerbate the accumulation of damaged proteins. Furthermore, the proteasomal E3 ligase complex is required for lifespan extension under reduced insulin signaling in C. elegans. Autophagy has also been implicated in aging. Genetic studies in C. elegans and Drosophila suggest that autophagy mediates, at least in part, the effects of insulin signaling and dietary restriction on longevity. It should also be noted that, although a drop in bulk protein turnover emerges as a common theme coupled with the aging process, not all proteins are uniformly affected, and individual protein levels do not always parallel the global trend. If the rate of protein synthesis is a determinant of aging, then manipulation of mRNA translation should have an effect on longevity. However, because protein synthesis is essential for growth and development, it is not straightforward to dissect its specific role in aging. Manipulation of general mRNA translation is likely to have pleiotropic effects, thus obscuring any explicit contribution to aging. Nevertheless, several recent studies capitalize on the genetic malleability of model organisms such as yeast, C. elegans, and Drosophila to investigate the link between protein synthesis and aging. These studies demonstrate that alteration of mRNA translation throughput has a significant impact on the aging process. Targeting a battery of key protein synthesis regulators reduces protein synthesis and extends lifespan. The process of protein synthesis involves three major, tightly regulated events: initiation of mRNA translation, elongation of the polypeptide chain, and termination of mRNA translation. Initiation is the rate-limiting step in mRNA translation and is the most common target of mRNA translation control. Global control of protein synthesis is generally achieved by changes in the phosphorylation state of initiation factors or their regulators. The rate of protein synthesis is mainly determined by regulation of two discrete steps during mRNA translation initiation: recruitment of the 40S ribosomal subunit at the 5' end of mRNA, and loading of the 40S ribosomal subunit with the initiator methionine tRNA. These events are coordinated by eukaryotic translation initiation factors, EIFs, EIF4E and EIF2, EIF2B, respectively. Phosphorylation of the EIF2 alpha subunit regulates dissociation of the EIF2B, EIF2 complex and subsequent EIF2 recycling. Similarly, phosphorylation of EIF4E binding proteins, 4EBPs, controls the availability of active EIF4E. The EIF4E is a key regulator of protein synthesis that recognizes the 5' end cap structures of most eukaryotic mRNAs and facilitates their recruitment to ribosomes. This is considered to be the rate-limiting step in translation initiation under most circumstances and is a primary target for translational control in many organisms. 5 EIF4E isoforms, IFE1 to IFE5, are encoded in the C. elegans genome. IFE1, IFE3, and IFE5 are expressed in the germline, whereas IFE2 and IFE4 are expressed specifically in somatic cells. Loss of IFE2 results in downregulation of protein synthesis in somatic cells and significant lifespan extension.